Good morning, everyone. So we have quite a number of people that have signed in, which is very gratifying. I'm Tom Sparks, and I'm the host for today. And I want to welcome all of you to this ACS webinar. We have a couple points to make before we get started. One is if you have any questions, there's a Q&A button at the bottom, and you can type in your questions anytime during the course of the webinar. And then when we get to the end, we'll go try and hopefully have enough time to get through all of them. The other aspect is that this webinar will be on the Agro website in about 2 weeks, thereabouts. So if you missed part of it or didn't get some of it, you should be able to get to it in a couple weeks from now. Everyone that is signed in will be on auto mute, hence the need to do the Q&A button to ask any questions of Steve as we go. So to get things going, again, I want to welcome all of you today, and it's my genuine pleasure to have Steve Duke present this webinar today. Steve has spent his last 45 years with the USDA ARS researching herbicide modes of action and resistance and natural products roles in plant protection and chemical ecology. He got his bachelor's degree at Henderson State College in biology, a master's at the University of Arkansas with a major in botany, and he got his PhD at Duke University in botany with a minor in biochemistry in 1975. And since 1975, he's been with the USDA ARS, initially at Stoneville, Mississippi, and then for the last 24 years or so at Oxford, Mississippi research unit, where he was leader of the group. He's had numerous awards and been very active in the American Chemical Society. He's past chair and fellow for the Agro Division. He is a fellow of ACS as well as the AAAS and the Weed Sciences Pathy Society and the International Weed Science Society. Among his many awards, and there's a lot, Outstanding Research Award from the Weed Science Society, the Molish Award for the International Allelopathy Society, and for the USDA ARS Mid-South Area Scientist of the Year, Outstanding Senior Scientist, and in 2017, the ARS Hall of Fame. So very well-rounded and has done a lot in his long career. On top of that, he's got 490 publications, including editor of a number of ACS symposium series. And so he's well qualified for the topic today, which is new herbicide modes of action for new commercial herbicides, searching for the Holy Grail. And on that note, Steve.
Thank you for the kind words, Tom. Can you hear me?
Yes, we can.
Okay, very good. I'll jump right into it. After a lag phase of a few years, there was a new mode of action herbicide introduced to the marketplace every 2 to 3 years for about 3 decades, from the 1950s until the early 1980s. And then the well went dry. You can see a little blip there at the end. We have had a couple of herbicides introduced, one introduced and one that will be introduced shortly in the last year or two. I'll discuss those in a bit more detail. But we've really had a paucity of new modes of action in the last 30 years or so. On this time course, I've marked the onset of evolved herbicide resistance, which began around the mid-1970s. Also, starting in the around 1990, there was a reduction in herbicide patent applications, and this really decreased dramatically after glyphosate-resistant crops were introduced in 1996 to a very low level. These patents that you see here mostly were herbicides that did not have new modes of action. Here you see the global increase in herbicide resistant weeds. And you can see, as I said, it began with a vengeance around 1975 and has continued linearly until the present. Some weed species are resistant to herbicides with up to 11 modes of action, and resistance to 7 modes of action can be found within a single population of some weed species. So farmers are running out of options in terms of using a new mode of action to manage herbicide resistance. This is something that Charles Darwin would have predicted early on with the heavy use of synthetic herbicides. Here you see the increase in glyphosate-resistant weeds worldwide. It's interesting. Glyphosate was introduced in 1974. It took over 20 years for glyphosate-resistant weeds to appear, but the same year that glyphosate-resistant crops were introduced, 1996, we had our first report of a glyphosate-resistant weed, not due to glyphosate-resistant crops, but they've come on very strong. And with the heavy use and heavy reliance on glyphosate, we have a lot of glyphosate resistance. We need new modes of action for resistance management. And we have situations like this. This is glyphosate-resistant Palmer amaranth in what was a cotton field in Georgia. You can see it's unmanageable. Another fine mess we've gotten ourselves into by not anticipating what was going to happen. There's some caveats. New modes of action aren't going to solve all of our problems because non-target site resistance, especially enhanced degradation capabilities, a compound with a new mode of action does not guarantee that it will be effective on existing herbicide resistant populations. Also, a compound with a new mode of action does not guarantee that it or a derivative will be a viable herbicide. There are a lot of reasons why simply finding a new compound that hits a new mode of action does not result in a commercial herbicide. And also, there is no cross resistance to some new herbicides with old modes of action, and there can even be negative cross resistance. And I'll mention this later, so that there can be new herbicides with old modes of action that can be used in resistance management. And my last caveat is, if I really knew how to find the Holy Grail, I wouldn't be giving this webinar. The number of modes of action for commercial pesticides is lower for herbicides than for the other two main categories of pesticides. This is from the Herbicide Resistance Action Committee, the Fungicide Resistance Action Committee, and Insecticide Resistance Action Committee. And this is partly due to as I showed you, the lack of new modes of action over the last 30 years, which has probably been exacerbated by glyphosate being so dominant and removing about half the value of the herbicide market, at least for a while. Herbicide Resistance Action Committee has a chart here of modes of action classifications. And here in one corner, you see unknown modes of action. So there are about 15 or 16 herbicides that are considered not where we really don't know the mode of action. These may be modes of action that are not like the ones that we know about. In fact, 3 modes of action that were very recently added to this chart came from this unknown mode of action category. One of them is synmethylin, which inhibits fatty acid thioesterases. This is quite an old herbicide, so it didn't really add a new herbicide to what was available to farmers, but now we know it has a new mode of action. This is indithol, an even older herbicide, which inhibits serine threonine protein phosphatase, and aclonophen was added. This is also a 30 or 40-year-old herbicide. Finally, the mode of action of it was found to be involved in synthesis of plastoquinone. A new mode of action for a new herbicide was added here. This is cyclopyramorate, which was developed by a Japanese company and added to the commercial herbicide market in Japan. This inhibits a new target site, and HRAC added another new herbicide that this is a dotted line because this compound is not actually on the market yet, but it's anticipated that it will be fairly soon. It's interesting that the cyclopyramorate and aclonophen were both herbicides that are involved in production of plasquinone, which is necessary for the activity of phytoene desaturase, which is the first enzyme totally dedicated to the synthesis of carotenoids. When you inhibit a carotenoid synthesis, you get a white plant, which of course dies. The enzymes that are in pink here, this is from a paper by Franck Dayan, are all involved in carotenoid synthesis, and these are the ones where we have commercial herbicides that attack these particular target site. As I mentioned, aclonophen was just discovered to inhibit this enzyme in the synthesis, and then this new herbicide was added that inhibits another new particular enzyme in the plasquinone pathway. So there are several approaches to discovery of new herbicides with new modes of action. The one that industries probably spent most of their time on is finding a new active compound and then determining its mode of action, hoping it'll be a new mode of action. I'm not going to talk about that. I'll talk about using natural phytotoxins as potential discovery process for finding a new mode of action, because many natural phytotoxins do have new modes of action. I'll also briefly discuss examining pharmaceuticals with targets not utilized by herbicides. And then I'll talk about finding out what target sites might be good targets for designing inhibitors that would be inhibiting an enzyme that is not a target site of currently used commercial herbicides. Let's start with natural products. Tom Sparks and his colleagues at Dow published several really nice reviews discussing this whole approach for discovery of all pesticides, not just herbicides. And Tom and I have a paper that will be coming out, should be online in a week or two, Journal of Ag and Food Chemistry, that discusses some of this in more detail, a rather long review on the successes in discovery of new pesticides from natural products. In one of these papers, Gerwick and Sparks, it was an analysis of what's been done showed that in the case of herbicides, here you see only 15% of the herbicides could have been discovered or were discovered from natural products. The number is higher for insecticides and fungicides. So we haven't been as successful using natural products in herbicide discovery as we have for the other pesticides. One of the real success stories is the discovery that the bottlebrush plant, which produces leptospermone and some other natural triketones, this compound was derivatized and modified to produce the first trikene ketone herbicides, which are all hydroxyphenylpyruvate dioxygenase inhibitors that inhibit plasquinone synthesis. So this was the last really big herbicide group from a natural product or any discovery process, and it was very successful. I might mention that these natural triketones like leptospermone are also HPPD inhibitors. Another big success story is that of L-phosphenothricin, a natural product from Streptomyces. The Streptomyces makes a tripeptide, which is sold as a bioherbicide in Japan. Synthesizing L-phosphenothricin, you get a racemic mixture, which is sold as glufosinate. And of course, this is the only herbicide that inhibits glutamine synthetase. Has been a very successful herbicide, but essentially inspired from the phosphenothricin from Streptomyces. This is one that could have been. We really don't know from the literature. Indothal, a very old herbicide, is a very close analog to cantharidin, and which is broken down to cantharidic acid in vivo. It only has 2 methyl groups different from indothal, and cantharidin is produced by blister beetles. This is the active ingredient in the centuries-old pharmaceutical Spanish fly. It's a very good herbicide itself, actually more herbicidal than indothal. So this could have been derived from this natural product. The structures of these 2 compounds were known back in the 1920s. So it's very possible indothal was derived from knowledge of these compounds. I'd like to point out that natural products from microbes may be better leads for new herbicides than leads from phytochemicals. That's because phytotoxic phytochemicals tend to be relatively weakly phytotoxic, primarily because autotoxicity is a problem in the evolution of phytochemicals that are phytotoxic. There are many microbial phytotoxins with novel modes of action that are more phytotoxic than most commercial herbicides, especially compounds produced by plant pathogens that kill the plant before they eat it. Some microbial phytotoxins, especially from plant pathogens, are plant and even plant species specific, and one of those I'll mention is tentoxin. Frank Dayan and I published a paper a few years ago which went through the modes of action of a great many of these natural compounds, especially microbial compounds. I'll just mention a few involved in amino acid synthesis. I mentioned phosphenothricin already, which inhibits glutamine synthetase. Phaseolatoxin has been known for years to inhibit ornithine carbamyl transferase required for production of arginine. Another microbial product is 5-methyltryptophan, which blocks the production of tryptophan. Gostatin inhibits the production of aspartate from oxaloacetic acid. Rhizobitoxin, a really old compound, inhibits beta-cystathionase, which is required for production of methionine. And all these compounds are quite phytotoxic. I mentioned tentoxin. It's a cyclic tetrapeptide from Alternaria alternata, which is a plant pathogen. It has excellent selectivity. It has at least 2 molecular targets, CF1 ATPase, and it inhibits the processing of the polyphenol oxidase into the plastid, which results in a yellow or white plant. This shows selectivity. This is Johnson grass with corn with soil applied tentoxin. You can see it kills Johnson grass, has no effect on corn. This is soybean and sickle pod. There's no taxonomic relationship between susceptibility and resistance to this compound, but serendipitously, it kills most of the important weeds in corn and soybeans. The only problem is it's a little too pricey to be used as a commercial herbicide. Efforts have been made to find derivatives that are simple to produce, but those have been unsuccessful efforts. Plant pathogens produce a number of different compounds that inhibit ceramide synthase, and these all cause accumulation of sphingosine bases. One of these is AAL toxin. It's one of the most phytotoxic compounds I've ever studied in the laboratory. This and these other compounds inhibit ceramide synthase, which can cause membrane dysfunction and program cell death, but we've found that accumulation of the precursors, these precursors are very phytotoxic and they can cause a membrane dysfunction and program cell death also. The only problem is these compounds are very toxic to mammals. So we tried to find compounds that would do the same thing that are not toxic to mammals, but so far unsuccessful. Actinonin is a streptomyces compound that inhibits peptide deformylase, which is required for protein formation in the plastid or chloroplast of plants. Quite active. It's a patented compound that nothing's developed from it commercially. Hydantocidin was discovered by Sankyo in Japan 30 years ago. There was a great deal of activity around this with various companies producing analogs, over 20 patents. It's a pro-herbicide that has to be phosphorylated in the plant to inhibit adenylosuccinate synthetase, which is a completely new mode of action. But despite all this effort among different companies, no commercial product was developed. Thaxtomin A is a unique cellulose synthase inhibitor. It was almost commercialized in a microbial bioherbicide by Marone Bioinnovations for use in rice and other cereals. It's from Streptomyces scabies, which causes potato scab. They got it as far as a trade name, but it did not make the marketplace. There's a great deal of excitement recently from results from a German lab. They found that a cyanobacterium produces this sugar, and you can see, at least in this bioassay, at the same concentration of glyphosate, it was more active than glyphosate. This is a shikimate pathway, which glyphosate inhibits EPSBS. Here, step 6, this sugar inhibits the enzyme that produces dehydroquinate earlier in the pathway. So it's inhibiting the same pathway, inhibiting production of aromatic amino acids. So there was a great deal of excitement about that. It's probably too early to see if anything will come of it. Another paper that generated some excitement was a paper from UCLA. This is aspartic acid, which was a known compound produced by a plant pathogen. You can see it's quite phytotoxic. And these people found that it inhibits an enzyme in the branched chain amino acid pathway other than acetolactate synthase, which is the target of a great many commercial herbicides. It's been very productive for the pesticide industry as a target site for herbicides. This particular compound inhibits dihydroacid dehydratase and is quite effective as a herbicide there. We'll see if anything comes of it. We published a short article mentioning that at least one company some years ago had found a number of synthetic compounds that inhibit this enzyme, but none of those were up to snuff to be commercialized. We've worked with a microbial bioherbicide, MBI 014, which is produced from a soil, which is a soil microbe. It has 2 phytotoxins in the preparation, romidepsin and spliceostatin C. And romidepsin, we published this last year, has a unique mode of action. It inhibits histone deacetylase in plant. You can see it's very active on Arabidopsis with a submicromolar I50. This is much more active than many commercial herbicides in the same bioassay. And the compound has to be reduced enzymatically in the plant to be maximally active as an enzyme inhibitor. This is all in this this paper in Journal of Natural Products published last year. I might mention that a lot of grasses produce benzoxanones like diboa, dimboa. Turns out diboa was found by a Spanish lab to be metabolically degraded to a compound called apo by soil microbes, and this is much more phytotoxic than the benzoxanoid allelochemicals. And a laboratory in Germany several years ago found that this compound, its mode of action is plant histone deacetylase inhibition. So this might be a good target site for herbicides, since we found two compounds, both romidepsin and this other lab found apo to hit this particular target. Spliciostatin C is an analog of spliciostatin A, which is a spliceosome inhibitor. Years ago, Monsanto patented herboxadiene, which is a rather complex microbial streptomyces product. It's also a splicing inhibitor. So we looked at these two, compared the two compounds, and found they were both very active on Arabidopsis. You can see the I50s here. And we examined their effect on both mRNA and protein production. Theoretically, if you're inhibiting the spliceosome here in the nucleus, you're ending up with the mRNA that contains introns that are normally spliced out by the spliceosome. So you get incorrect translation of the gene, resulting in inactive proteins that are translated and folded wrong. And in fact, some of our results are that both the compounds modified constitutive and alternate splicing. In several cases, the transcripts affected by both inhibitors had additional and longer transcripts, either by intron retention or by alternative 5' or 3' splicing sites. And even though herboxadiene had higher phytotoxicity, the proteins and genes affected were a bit different than those affected by spliciostatin C. This is unpublished as of yet. There are quite a few intriguing microbial phytotoxins that we have clues that have unknown molecular targets. One of these is a compound from Mycosynthetics, which was being developed by Dow, mevallocidin, produced by several microbes. It translocates and causes bleaching. It's fluoromobile, and it may be an inhibitor of mevalonate kinase. Another one is pseudocidin that Dow worked on. It's produced by a microbe. It's an analog of jasmonic acid, and like coronitine, it's quite phytotoxic and may be interfering with jasmonic acid, which is a natural plant hormone. Muscalitox neglycone is a natural compound, a small polyamine from a microbe that was being looked at as a potential mycoherbicide in Italy. We looked at its potential mode of action. You can see here that it has a very low submicromolar I50, which is again much higher activity than many commercial herbicides. Along with BASF, we did metabolomics assays to look at its effect on especially amino acid metabolism. It really messes up amino acid metabolism. And we looked at one predicted target site, alanine aminotransferase, and found no activity. We concluded that it may inhibit one or more aminotransferase not examined. It may affect amino acid transporters, or it may be a protoxin that, so we weren't looking at its effect on the target site as it would be acting in vivo. those are some natural product clues and leads that are out there. Screening pharmaceuticals, especially pharmaceuticals for infection diseases, might possibly lead to new target sites. The laboratory of Josh Milne in Perth, Australia, is one that is using this approach. Here's a compound that is used as a pharmaceutical. It inhibits DNA gyrase. It's quite phytotoxic, and they've been looking at analogs of this as potential herbicides. This is the biochemical pathways of Plasmodium, the malaria microbe. You notice it has a organelle, the apicoplast, which is a vestigial chloroplast. It's hard to believe, but Plasmodium evolved from a microbe that had a chloroplast. And many of the target sites in the apicoplast are identical to target sites in plants. In fact, glyphosate is effective on Plasmodium, although I don't think you'd want to drink Roundup herbicide to cure yourself of malaria. One compound that we've looked at, and people have looked at several others, this is the most important anti-malarial drug at this time. It's actually a plant product, artemisinin, a sesquiterpene produced by only one plant species. It's quite active against Plasmodium, but it's also a very highly active phytotoxin. The mode of action in Plasmodium is still not, I don't think, completely worked out, but it appears to covalently bond certain proteins, something we predicted back in the 1980s, and it's probably doing the same thing in plants. Quite active. At least one company was quite interested in it as a lead for herbicide some years ago. The problem is it's rather expensive even as a pharmaceutical, but it is a new mode of action. There are two previous approaches to target site discovery. That is, what is a good target site? One is to find inhibitors of enzymes of a pathway for which there are good herbicides, such as a shikimate pathway and the branched chain amino acid pathways, two that I've shown you already, and then look for good in vitro enzyme inhibition compounds. And that hasn't been very productive. The other is to evaluate gene knockouts and to determine whether a particular target might be good as a target site for herbicide. Gene knockouts generally result in 100% inhibition, whereas herbicides probably only inhibit, at least a good herbicide, probably only inhibits a portion of the enzymatic activity. So partial knockouts would be better at predicting effects of herbicides. So finding good herbicides for new identified target sites has been quite problematic. In other words, starting with a target site and trying to find a good inhibitor. Simply because a herbicide inhibits a target site not found in humans is not necessarily a good rationale for picking a target site. These are the commercial herbicide targets not found in humans. However, quite a few commercial target sites are found in humans, and these particular 4 target sites are very important target sites for commercial herbicides. This particular one I have in orange is a new target site that will probably hit the market very shortly. Frank Dayan and I published a paper last year in which we discussed 2 rationales for picking a particular target site to try to find inhibitors. One is finding target sites that result in accumulation of toxic metabolites, and the other is to find target sites that are in very low abundance. Let me explain. So if we have a target site, let's say here, that by inhibiting it, we result in this metabolite that you find in very low abundance increasing dramatically. It might be because this particular low abundance target site is kept metabolically very small because it is toxic. And I'll give you some examples of this that we know about. Perhaps the best, so inhibition, in this case, inhibition of a relatively small percentage of the enzyme will still cause phytotoxins to accumulate. Probably the prime example of this are the PPO inhibitors, protoporphyrin oxidase inhibitors. The herbicide arrives at the plant surface, hits the target site, which is a plastid envelope. In this case, we inhibit the target. In this case, protoporphyrin 9 is produced by the enzyme. In this case, the substrate, instead of accumulating, leaves the enzymatic target site, accumulates in the cytoplasm. It's then oxidized by a non-PPO enzyme, probably a peroxidase, to protoporphyrin 9, which in the presence of molecular oxygen and sunlight produces toxic oxygen species, singlet oxygen mainly, which results in a cascade of oxidative degradation products and very rapid killing of the plant. PPO inhibitors are the really good ones, are active at very low concentrations, and this is probably the reason why, because you only need to inhibit a small amount of the protoporphyrinogen, not protoporphyrinogen or the PPO, to cause accumulation of protogen outside the chloroplast. In this case, we're getting accumulation of the product, but the same thing could happen with accumulation of the substrate. And there is evidence that accumulation of alpha-ketobutyrate, 2-aminobutyrate quinate with ALS inhibitors contributes to their toxicity. Accumulation of quinate accounts for part of the toxicity of glyphosate. Accumulation of as I mentioned, sphingoid bases contribute greatly to the toxicity of ceramide synthase inhibitors, and accumulation of some metabolic intermediates accumulate in response to glufosinate result in its toxicity. So this is another approach. The enzymes here in this pathway are the arrows. A enzyme in very low abundance might require less herbicide to inhibit it. So is this a preferred target? I'll give you one example that suggests that this might be the case, and that enzymes in high abundance have not been picked because too much herbicide is required to kill a plant with these high abundance proteins. In the case of glyphosate, which is a really relatively high use rate herbicide, if we had to use much more glyphosate than is used, it might not have been picked as a commercial herbicide. And in fact, one mechanism of resistance is gene amplification, where we get many more copies of the ESPS gene than found in the wild type, and in which cases we have a lot more of the enzyme and much higher doses of herbicide have no effect. This is pigweed with these high levels of the enzyme. It simply dilutes out the herbicide. And we're finding other cases of gene amplification for other herbicide classes. So if EPSPS had been present in the same levels that we see in these gene amplified cases of weeds, it would have never been developed as a herbicide. So there may be cases where there are genes and enzymatic sites that are not appropriate as herbicides because of the level of the enzyme that's available. I mentioned earlier that old chemistries too might yield new modes of action. These are all diphenyl ethers. Where we have commercial cases are PPO inhibitors, there are quite a few PPO inhibitor diphenyl ethers. There are quite a few ACCase inhibitor diphenyl ethers. We have the one solanyl diphosphate synthase inhibitor that was just discovered, an old diphenyl ether. Nitrophin, there's evidence from Carl Kunert and Peter Berger's lab from years ago that it inhibits plasmid ATPase. So we have probably at least 4 modes of action with this basic chemical type. Frank Diane published a paper a few years ago showing that triclosan, a diphenyl ether that was in toothpaste until 2019 when it was banned, is a very good phytotoxin that inhibits enol reductase, which is involved in plant fatty acid elongation. And this particular plant or microbially produced phytotoxin, cyprin, hits the same target site. So here we have 5 modes of action among diphenyl ethers. Are there more? And there may be other chemical classes that would yield more than one new mode of action. This suggests too that with the same chemical class, you might be able to develop a herbicide that has 2 modes of action in the same compound, something that Johnny Gressel in a recent paper in Pest Management Science has been promoting as a way of fighting evolved target site herbicide resistance. There's several obstacles for effective herbicides with new modes of action. Most of these obstacles are the same obstacles for any pesticide: intellectual property issues, human and environmental toxicology issues. I mentioned the ceramide synthase inhibitors are really toxic to mammals, even though they're fantastically active phytotoxins. Cost of production, a lot of these natural products are very costly, even as pharmaceuticals. So simplifying them and reducing the cost is imperative in the case of many of these. And market niche. You can have a great herbicide that may be quite active, but if it's not competitive with what's already out there, this is a changing target as resistance evolves. But this is a consideration when looking for new modes of action also. So there are no clear blockbuster herbicides with new modes of action on the horizon. Is there a holy grail out there? I think there probably is. Let's hope we're more successful than these fellows were. A positive parting thought is that there's considerably new interest and investment in a new herbicide discovery by old and new approaches, both large and small companies. As I mentioned, there's a new herbicide here, tetflupyrolimet, which FMC company should introduce to the market very soon, which has a totally new molecular target site. It inhibits dihydroorotate dehydrogenase, which shuts down pyrimidine synthesis. There's some new companies with new discovery technologies, such as artificial intelligence and strategies working on herbicides with new modes of action. And these small companies are, in some cases, using approaches and strategies that, as far as I know, aren't being matched by some of the larger companies. So let's hope they're able to contribute to the introduction of new modes of action in the near future. Another positive parting thought is another technology that's coming on very fast and will impact all of commercial herbicides is precision agriculture that directs herbicides only to the weeds, should allow the economical use of expensive products, making some high-cost molecules viable as herbicides. It's hard to predict what the impact of this will be, but it probably could make some of these high-cost compounds that are not viable now viable if you can only spray the desired vegetation with them. I'd like to thank the people that were involved in the production of the data that came from our lab. And thanks for listening. If you have any questions, Tom can relay them to me now.
Well, Steve, thank you very much for a really excellent overview of this really cool topic. There are a number of questions that are we've been submitted, and I have a few myself. Going back to the first part of your talk, what do you think is the main reason for lack of new modes of action after the 1980s?
I published a paper on this back in 2012, and there are a couple of reasons. I think a real main reason is the fact that glyphosate-resistant crops greatly diminished the value of the herbicide market, and the major companies stopped a lot of their discovery effort. It went down. You can see the number of patents went down. It was already going down to some extent. I think probably because at that point, resistance wasn't that big a deal, and farmers had tools for almost everything already. So, and consolidation of industry probably has played some role also. Consolidation of industry meant fewer groups were using their fewer strategies for discovery, so there was less diversity in thought in discovery efforts.
All right, thanks. One of the questions that's been submitted, many of the microbial toxins hit target sites in the metabolic pathways that are not unique to plants. So obviously there's going to be some non-target effects. And I guess part of the question would be, are these going to require the development of resistant plants, crop plants, in order to implement these?
Not necessarily. I gave a list of target sites that have human targets that are the same. And for most of those targets, there are no transgenic herbicide resistant crops. There are glufosinate resistant crops, but glufosinate was used to some extent before those crops were introduced, actually a year before glyphosate resistant crops. But there are no mitotic inhibitor herbicide resistant crops. In the case of mitotic inhibitors, the ones that are most active on humans are not very good herbicides, and the ones that are most active as herbicides have very little effect on mammals. So the toxicity issue is not a big deal. So yeah, it's not a requirement. Not a requirement.
All right. Go on.
HPPD inhibitors, we do have HPPD inhibitor resistant crops coming on, but HPPD inhibitor herbicides are used extensively. Before then, I might mention that one pharmaceutical that's used for individuals that have tyrosine metabolism defects is virtually identical to a commercial HPP inhibitor triketone. So apparently there's been no toxicology issue though in using those herbicides.
All right. One other question that came through was, what was and if you know this one, what has been the key reason for the unsuccessful attempts to make hydantocide a commercial product?
I really don't know. I mean, these are the sorts of things that companies are pretty mum about. But there was a great deal of excitement for about 5 years, and then the well went dry. No more patents, not much interest anymore. I don't know whether it's market niche, toxicology issues. It could have been any of the things that I mentioned as detriments to commercialization.
Okay. Another question that came through is you said that good herbicides will inhibit only partially an enzyme versus gene knockdown. What do you mean by this? A herbicide to be a good um, a herbicide to be good does has to hit 100% of activity or not?
It, no, what I
I
was
think
trying to point out is that I think that, and I firmly believe this, even though there are really no good data to prove this, that to be a really good herbicide, it should kill the plant without inhibiting all of the enzyme in the plant. PPO
but
inhibitors are good examples of herbicides that are probably only inhibiting a fraction of the PPO that's actually in the plant leaf, simply because if you inhibit any of it, you're going to get protoporphyrin 9 accumulating, and that's going to be, if you get enough protoporphyrin 9 accumulation, you're going to kill the plant. There are other cases, perhaps ALS inhibitors, that you probably have to inhibit more of the enzyme, but probably not all of it. Saturating the enzyme in the plant is going to be very difficult. But if you can inhibit half the enzyme that's there or a third of the enzyme and kill the plant, that's highly desirable. But as I said, there are very few data on how much of these enzymes are actually in plants or target plants, and how much is actually physically in contact with the herbicide in a dying plant. So this is something that's a little theoretical, but I firmly believe that it's highly desirable that you look at targets that don't require all of the target to be inhibited to kill the plant.
all right. So if you had something that was 100%, that wouldn't be a bad thing.
It wouldn't be a
I
bad thing. I think it's just probably logistically very difficult to get 100% saturation in the plant.
all right. Another question that came through is what type of herbicide, from your point of view, was most desirable for a company to develop? A broadleaf, grass, non-selective? What would you shoot for?
Huh. That's really out of my wheelhouse. Uh, yeah, that's a more practical question than a field person. Uh, there are many people other than me that could best. That's a market niche question that can make a weak herbicide very desirable, or a strong herbicide not very desirable, depending on what products are already out there for particular market niches.
Okay, another question that came through is what is your opinion on Protax? This is all capitalized as a new synthetic modality in the area of herbicide discovery.
Uh, Protax, I don't know.
Okay. Another question, do you think that increasing the use of bioherbicides will reduce the value of synthetic herbicides with new modes of action? Question mark.
Well, if there is a large increase in bioherbicides, certainly it would reduce the value of practically at least some commercial synthetic herbicides. When we talk about holy grails, finding a really good bioherbicide is even more of a holy grail than finding a new mode of action for a synthetic herbicide. There just really aren't any good bioherbicides out there. They're good bioinsecticides and good biofungicides, but there's no really good bioherbicides. So that's even a bigger hill to climb than finding a new mode of action. So I don't see any of those hitting the marketplace in the near future. So, and I wouldn't bet on bioherbicides becoming a major part of the herbicide market in the next decade or so.
Okay. Another question that came through is what makes a target protein ideal for an herbicide? And do you have a favorite target in plants?
Well, PPO has been my favorite target because we work on the mode of action of that for several years. Very interesting mode of action and unique mode of action. It's gotten more and more complicated and more interesting as it's gone, and resistance to it is also quite interesting. But that doesn't necessarily make it the most desirable target site.
All right, there's been a whole bunch of questions that have come through, so I'm not sure we're going to get through all of these, so I'm kind of having to pick and choose. An interesting one is, what is the potential of RNAi technology for weed management, in your opinion?
Oh, that's a good question. Monsanto spent a fair amount of effort in that area, and with using RNAi for weed management. And in the case of weed management, you've got to spray it like a herbicide instead of having the crop produce the RNAi like you do for insects or pathogens. So getting it into the weed at economical levels and effective levels is a challenge. Nothing's been published on this. And I don't see it being developed at this point. But I may be unaware of development that's going on. But I haven't seen anything on it in quite some time. I know more than I can say, but I'm not aware of anything going on at this time. It's a challenging approach.
All right. Going back to your comments about target driven discovery, which from the get-go, most everybody would think that if you know what the target site you're going after is, it should actually be a reasonable approach. And so the question is, why do you think target site herbicide discovery, i.e. In vitro screening, has not been successful in our industry? What could be done differently to increase the translation rate from in vitro to whole plant activity?
I've never been involved in that approach. My colleagues that have been involved in that approach have told me that the biggest problem is that is finding a really good inhibitor of the enzyme that also has the right physicochemical properties to be a good herbicide. That is something that's readily taken up and translocated. I mentioned the branched chain amino acid pathway. I know American Cyanamid spent quite a bit of effort looking at all the enzymes in that pathway, since they had very good inhibitors for acetolactate synthase, or AHOS as they called it. And they got good inhibitors of the enzymes, but they never got a commercial herbicide for carry, the next enzyme after ALS, or the enzyme that aspartic acid inhibits. They had inhibitors for that, and none of those were developed. So finding a compound that is both a good in vitro inhibitor that has the physicochemical properties that are required for a compound that can be used economically as a commercial herbicide has been very difficult. And it also may be that some of the target sites picked were difficult for some of the reasons, or not as good for one of the two reasons that I mentioned, that I thought a target site might be preferable simply because inhibiting it causes some toxic intermediates to accumulate, or that it's present in very low amounts in vivo.
All right, I think we've got time for one last question. We can squeak it in. And again, there's been a whole bunch, but this one is, any advancement in application technology to make herbicides more bioavailable?
Well, as far as formulation goes, not that I'm aware of. As far as application technology goes, as I mentioned, I think people are looking forward to precision agriculture methods where you have a computerized spray rig that can separate the weeds from the crop and spray only the weeds, and preferably even differentiate between different weed species so you're applying the proper herbicide to the appropriate weed species. Theoretically, that's all doable. It's coming on quicker now than I thought it would several years ago, but it is. I see papers on this topic. People are working on it very hard, and I expect eventually this will impact the herbicide market. Both the herbicides that are already there, it may impact the economics of herbicides that are already there by, if farmers are using a lot less of them, that may be a concern. Whereas a herbicide that's too expensive to be used now might be economical with that sort of application technology.
Thank you very much, Steve. And with that, we've basically run out of time today. I want to thank everybody that tuned in, and I want to thank the people that submitted questions. Thank you for that. We'll see about trying to get answers to those that we weren't able to get to today. And with that, thank you all. And again, thank you very much, Steve, for a really excellent seminar.
Thank you.